Selamat datang di UPT Laboratorium Penelitian Terpadu Universitas Sumatera Utara. UPT Laboratorium pada pelayanan dengan mengutamakan kepentingan penelitian di lingkungan Universitas Sumatera Utara yang meliputi fakultas dan program studi, lembaga penelitian, pusat unggulan iptek, dan satuan kerja lainnya. Tugas pokok UPT Selamat datang di UPT Laboratorium Penelitian Terpadu Universitas Sumatera Utara. UPT Laboratorium Penelitian dan Kerjasama. UPT Laboratorium Penelitian Terpadu USU juga pada pelayanan dengan mengutamakan kepentingan penelitian di lingkungan Universitas Sumatera Utara yang agar dapat menghasilkan dan produk yang bersifat inovatif dan berhasil dimonetisasi unggulan iptek dan satuan kerja lainnya. Tugas pokok Selamat datang di UPT Laboratorium Penelitian Terpadu Universitas Sumatera Utara. UPT Laboratorium Penelitian dan Kerjasama. UPT Laboratorium Penelitian Terpadu USU juga pada pelayanan dengan mengutamakan kepentingan penelitian di lingkungan Universitas Sumatera Utara yang agar dapat menghasilkan dan produksi bersifat inovatif dan berhasil dimonetisasi unggulan iptek dan satuan kerja lainnya. Selamat datang di UPT Laboratorium Penelitian Terpadu Universitas Sumatera Utara. UPT Laboratorium Penelitian dan Kerjasama. UPT Laboratorium Penelitian Terpadu USU juga pada pelayanan dengan mengutamakan kepentingan penelitian di lingkungan Universitas Sumatera Utara dan standar dan kerjasama siswa S1 inovatif dan berhasil Laboratorium Penelitian Terpadu USU dan satu lagi Anjelisa Laboratorium. Selamat datang di UPT Laboratorium Penelitian Terpadu Universitas Sumatera Utara. UPT Laboratorium Benda secara kualitatif. UPT Laboratorium Penelitian Terpadu USU juga pada pelayanan dengan konsentrasi logam dan senyawa pada sampel organik maupun anorganik. Laboratorium Uji Sifat Mekanik melayani pengujian sifat-sifat mekanik suatu bahan atau material seperti kekuatan tarik, kekuatan tekan, dan kekuatan lengkung, serta penghalusan sampel hingga berukuran nanometer. Laboratorium Uji Sifat Termal melayani pengujian sifat-sifat termal pada suatu material. Laboratorium Renewable Energy melayani simulasi pengkonversian energi dan yang berkaitan dengan energi baru terbarukan. Laboratorium Biologi Bahan Molekuler melayani penelitian, preparasi, dan ekstraksi sampel. Dan juga UPT Laboratorium Penelitian Terpadu USU juga memiliki alat-alat penunjang penelitian seperti Scanning Electron Microscopy atau SEM, Atomic Absorption Spectrometry atau AAS, Universal Testing Machine atau UTM, Ball Mill, Siftronic Melting Point, neraca digital, furnace, fuel cell, generator solar panel, wind turbine, PLC, humidifier, sentrifugasi, sonicator bed. UPT Laboratorium Penelitian Terpadu USU melayani pengujian karakterisasi sampel untuk mahasiswa, dosen, peneliti baik dari dalam maupun luar USU, dan juga memberikan kesempatan penelitian menggunakan tempat dan fasilitas di UPT Laboratorium Penelitian Terpadu USU.
Saya Ramadhani Banuria, di sini di UPT Laboratorium Penelitian Terpadu USU. Saya sebagai Kepala Bidang Pelayanan dan Promosi. Tugas di sini membantu tugas daripada Kepala UPT Laboratorium Penelitian Terpadu, merencanakan pengembangan laboratorium semua bidang dengan bekerjasama dengan Kepala Laboratorium. Kemudian mengkoordinasi seluruh kegiatan pengujian yang dilaksanakan di laboratorium.
UPT Laboratorium Penelitian Terpadu memiliki beberapa grup penelitian. Grup penelitian yang sedang berjalan saat ini di UPT Laboratorium Penelitian Terpadu adalah Green Concrete, IoT, atau Internet of Things, Utilization of Agricultural Waste, sodium ion battery, bioenergy, microplastic and heavy metal, material isolator, renewable energy agriculture, functional food and biomolecular system. Sebagai salah satu wujud pengabdian UPT Laboratorium Penelitian Terpadu kepada masyarakat, UPT Laboratorium Penelitian Terpadu membuka peluang kepada instansi pemerintahan negeri atau swasta maupun masyarakat umum untuk melakukan kerjasama. Di 2021 ini, UPT Laboratorium Penelitian Terpadu sedang menjalankan kerjasama penelitian dengan pihak perusahaan listrik negara atau PLN terkait penelitian faba.
Pada tahun 2021, UPT Laboratorium Penelitian Terpadu menjalin kerjasama dengan perusahaan listrik negara tentang penelitian pemanfaatan faba. Selain itu, di tahun 2021 juga UPT Laboratorium Terpadu sudah membuat inovasi tentang pembuatan pakan ternak dan juga hand sanitizer. Pada tahun 2020, UPT Laboratorium Penelitian Terpadu telah menghasilkan beberapa luaran, di antaranya publikasi internasional sebanyak 23 paper, publikasi nasional 3 paper, dan paten granted 7.
Saya Wakil Siregar. Di UPT Penelitian Terpadu, saya bertugas sebagai Kepala Bidang Penjaminan Mutu dan Penggunaan Teknologi Informasi. Adapun target kami dalam waktu dekat ini adalah agar beberapa jenis pengujian yang dilayani oleh Penelitian Terpadu mendapatkan akreditasi ISO IEC 17025-2017. Dengan adanya akreditasi bertaraf internasional ini, diharapkan tingkat kepercayaan konsumen untuk melakukan pengujian meningkat, dikarenakan memenuhi beberapa aspek kualitas, antara lain ketidakberpihakan, kerahasiaan, keakuratan alat uji, kompetensi penguji, pemilihan, verifikasi dan validasi metode, serta pengendalian data dan manajemen informasi.
Kami anggota dari UPT Laboratorium Penelitian Terpadu USU siap menjalin kerjasama untuk membantu penelitian Anda, dan kami siap mengabdi pada masyarakat untuk memajukan bangsa Indonesia.
UPT Laboratorium Penelitian Terpadu USU.
Coba,
Dengar kan suara? Saya
minta serta buka kamera ya.
Before we start this webinar, please turn on your camera for all participants. Dengar. Udah, Bang. Assalamualaikum warahmatullahi wabarakatuh. Good morning and welcome everyone.
Halo? Halo? Bisa kedengaran?
Kok mau dengar apa?
Halo? Halo? Kedengaran nggak? Halo? Bisa?
Iya, iya. Ya, Kak. Udah, Kak. Good morning and welcome everyone. Thank you for joining us. I am Raisa Zahra and Miss Veryani Gusniesti as M.Hum, and we will be your MC today. Our topic today is Introduction of Antimicrobial Resistance Research, focusing on people and the activities of the AMR Research Center. This webinar will be brought in 3 languages, English, Bahasa, and Japanese. Now let's begin this webinar with Indonesia National Anthem, Indonesia Raya, and followed by a prayer from Mr. Febri Rizky, S.Si.
それでは早速ですが、本日初めにインドネシアラヤというインドネシアの国歌をお聞かせていただきます。その後、フェブリリス様からお祈りのリード。
Indonesia
Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Let us bow our head for a moment, pray to God, Allah Subhanahu wa Ta'ala, so that our webinar can be precious to all of us. I will lead this prayer based on the teaching of Islam, and those who are not Muslims, you are pleased to pray according to each of your belief. Ya Allah, today in this webinar, we are gathering here to hold this webinar. May this webinar be a useful science assembly, be a medium of sharing useful ideas, knowledge, and experiences of scholars, researchers, and students of various disciplines. May the webinar we organize today benefit to our life, broaden our knowledge, science of ideas, and lead us to be successful, productive people, which in turn will boost dignity of our nation. Ya Allah, you are the one who can fulfill our prayer. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina azabannar. Izzati subhana rabbika rabbi amma yasifun. Wassalamu alal mursalin walhamdulillahi rabbil alamin. Amin.
Thank you, Mr. Febri. May Allah always guidance us. The next agenda is welcoming speech from Head of Integrated Research Laboratory of Universitas Sumatera Utara. Please welcome Insinyur Rahmi Karolina ST.MT.
Terima kita semua teradai mau sogo thank you jo chokarano kange speech desu. それではラミカロリナ先生、どうぞよろしくお願いいたします。
Thank you very much, Ms. Raisa and Ms. Veryani. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. The Honorable Professor Popi Anjelisa, as Vice Rector 3, Universitas Sumatera Utara. The Honorable Professor Motoyuki Sugai, the Director, Anti-microbial Resistant Research Center, Japan. Ohayou gozaimasu. I would like to welcome all of you to our 6th webinar series, Top Scientists. Thank you for being here with us today. I will also express my sincere appreciation to the Integrated Research Laboratory crew, especially Mr. Adrian. As the head laboratory of Molekuler Biology and Integrated Research Laboratory Universitas Sumatera Utara. Thank you so much for leading your team to create this webinar. We would not have been here in this beautiful morning without you and the team. And especially honored to welcome the keynote speaker in this webinar, Professor Motoyuki Sugai from Anti-microbial Resistant Research Center, National Institute of Infectious Disease, Japan. In today's webinar, our agenda is to focus on antimicrobial resistance on people and also giving us some activities of the Antimicrobial Resistant Research Center, which we can learn from that and also apply to our laboratory. I hope that through this event, we all can learn to be good and critical thinker. So I wouldn't like to take too much of your time, so I will hand it over to M.Si, Ms. Raisa, and Ms. Veryani to proceed to the next agenda. Once again, welcome and thank you very, very much for attending this webinar. I wish all participants have gained knowledge and have fun. Thank you very much. Arigato. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for your wonderful opening speech. The next agenda is opening speech from Vice Rector 3 of Universitas Sumatera Utara. Please welcome Profesor Dr. Popi Anjelisa Zaitun Hasibuan, S.Si, M.Sc, APT.
続きまして、北
Thank
島
you.
原大学の副学長からの開会の挨拶です。それでは、ポピー・アンジェリーサ・ザイトン・ハシブン先生、どうぞよろしくお願いいたします。
Thank you. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Thank you to each and every one of you for being here today at this wonderful place. Honorable Mrs. Rahmi Karolina, Honorable Professor Motoyuki Sugai, and all participants in the morning. It is such an honor for me to speak as Vice Rector 3 Universitas Sumatera Utara. Let me begin by giving you a warm welcome to the Top Scientist webinar. Before we get started, I would like to express my appreciation to the UPT LPT crew who generously helped us make this webinar come true, Mrs. Rahmi Karolina and the rest of the UPT LPT crew. We could not have done it without you. And the best appreciation to Professor Motoyuki Sugai from Antimicrobial Resistant Research Center, National Institute of Infectious Disease, Japan. Give us your precious time to share your best experience. In today's webinar, we will learn all about antimicrobial resistance and show us to the activity in Research Center, National Institute of Infectious Disease. I hope this webinar can provide very useful information and knowledge to all of us. Well, I do not want to take too much of your time. Now, by saying Bismillah, Top Scientist webinar on 18 November 2021 officially open. Thank you. It is great to see so many of you here. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Prof. Next agenda is for our documentations of today's event. We would like to take pictures by taking screenshots. We request everyone to turn on the camera.
今日のイベントのドキュメンタリーとしてスクリーンショットを撮りたいと思います。皆様のズームカメラはオンにしていただきますと幸いです。
Alright. 1, 2, 3. Once again, 1, 2, 3. Once again. All right, 1, 2, 3. Thank you. Before I hand over the mic to the moderator, I would like to inform all of you that this webinar is broadcast live on our YouTube channel, Lab Terpadu USU. And if you have any question during the presentation, just type the question into the chat box and just write freely in Bahasa, Japanese or English, and we will translate it later. Because after the presentation, we will have a Q&A session.
今回のウェビナーは Lab Terpadu USU という YouTube チャンネルにリアルタイム配信で行います。また、講演後に質疑応答の時間を設けております。菅井先生にご質問がありましたら、英語、インドネシア語、または日本語でチャットにご質問をご記入ください。もしインドネシア語で質問したら、私たちは英語か日本語に翻訳します。
Selamat datang di UPT Laboratorium Penelitian Utara. UPT Laboratorium Penelitian Terpadu USU merupakan unit pelaksana teknis yang berorientasi pada pelayanan, dengan mengutamakan kepentingan penelitian di lingkungan Universitas Sumatera Utara yang meliputi fakultas dan program studi, lembaga penelitian, pusat unggulan iptek, dan satuan kerja lainnya. Tugas pokok UPT Laboratorium Penelitian Terpadu USU adalah menyelenggarakan kegiatan laboratorium seperti pelayanan, penelitian, dan kerjasama. UPT Laboratorium Penelitian Terpadu USU juga mengakomodasi kepentingan lembaga inovasi dan inkubasi bisnis teknologi USU dengan tujuan agar dapat menghasilkan produk-produk yang bersifat inovatif dan berhasil dimonetisasi.
Di sini kita ada pengujian SEM, kemudian ada pengujian AAS, ada pengujian uji mekanik, kemudian ada juga ball mill, kita juga punya centrifuge, dan juga ada oven furnace yang suhu temperaturnya bisa mencapai 1200 derajat. Kemudian kalau untuk pelayanan, sebenarnya kita juga menerima pelayanan untuk mahasiswa S1, S2, dan S3 untuk melakukan penelitian. Jadi rate-nya bisa penelitian di sini dalam 1 bulan, 3 bulan, ataupun selama setahun.
UPT Laboratorium Penelitian Terpadu USU memiliki 1 kepala unit, 3 kepala bidang, 6 kepala lab, 10 laboran, dan 3 staf penunjang administrasi. Laboratorium Penelitian Terpadu USU memiliki 6 jenis laboratorium. Laboratorium karakterisasi struktur bahan, melayani pengujian topografi dan morfologi dari permukaan benda secara kualitatif. Laboratorium uji sifat kimia, melayani pengujian dan penentuan sebagai konsentrasi logam dan senyawa pada sampel organik maupun anorganik. Laboratorium uji sifat mekanik, melayani pengujian sifat-sifat mekanik suatu bahan atau material, seperti kekuatan tarik, kekuatan tekan, dan kekuatan lengkung, serta penghalusan sampel hingga berukuran nanometer. Laboratorium uji sifat termal, melayani pengujian sifat-sifat termal pada suatu material. Laboratorium renewable energy, melayani simulasi pengkonversian energi dan penelitian yang berkaitan dengan energi baru terbarukan. Laboratorium biologi bahan molekuler, melayani penelitian, preparasi, dan ekstraksi sampel. Dan juga UPT Laboratorium Penelitian Terpadu USU juga memiliki alat-alat penunjang penelitian seperti Scanning Electron Microscopy atau SEM, Atomic Absorption Spectrometry atau AAS, Universal Testing Machine atau UTM, ball mill, Siftronic Melting Point, neraca digital, furnace, fuel cell, generator solar panel, wind turbine, PLC, humidifier, sentrifugasi, sonicator bed. UPT Laboratorium Penelitian Terpadu USU melayani pengujian karakterisasi sampel untuk mahasiswa, dosen, peneliti baik dari dalam maupun luar USU, dan juga memberikan kesempatan penelitian menggunakan tempat dan fasilitas di UPT Laboratorium Penelitian Terpadu USU.
Saya Ramadhani Banuria, di sini di UPT Laboratorium Penelitian Terpadu USU, saya sebagai Kepala Bidang Pelayanan dan Promosi. Tugas di sini membantu tugas daripada Kepala UPT Laboratorium Penelitian Terpadu, merencanakan pengembangan laboratorium semua bidang dengan bekerjasama dengan Kepala Laboratorium. Kemudian mengkoordinasi seluruh kegiatan pengujian yang dilaksanakan di laboratorium.
UPT Laboratorium Penelitian Terpadu memiliki beberapa grup penelitian. Grup penelitian yang sedang berjalan saat ini di UPT Laboratorium Penelitian Terpadu adalah Green Concrete, IoT, atau Internet of Things, Utilization of Agricultural Waste, Sodium-ion Battery, Bioenergy, Microplastic and Heavy Metal, Material Isolator, Renewable Energy Agriculture, Functional Food and Biomolecular System. Sebagai salah satu wujud pengabdian UPT Laboratorium Penelitian Terpadu kepada masyarakat, UPT Laboratorium Penelitian Terpadu membuka peluang kepada instansi pemerintahan negeri atau swasta maupun masyarakat umum untuk melakukan kerjasama. Di 2020-21 ini, UPT Laboratorium Penelitian Terpadu sedang menjalankan kerjasama penelitian dengan pihak perusahaan listrik negara atau PLN terkait penelitian faba.
Pada tahun 2021, UPT Laboratorium Penelitian Terpadu menjalin kerjasama dengan perusahaan listrik negara tentang penelitian pemanfaatan faba. Selain itu, di tahun 2021 juga UPT Laboratorium Terpadu sudah membuat inovasi tentang pembuatan pakan ternak dan juga hand sanitizer. Pada tahun 2020, UPT Laboratorium Penelitian Terpadu telah menghasilkan beberapa luaran, di antaranya publikasi internasional sebanyak 23 paper, publikasi nasional 3 paper, dan paten granted 7.
Saya M.Sc Sugai. Di UPT Penelitian Terpadu, saya bertugas sebagai Kepala Bidang Penjaminan Mutu dan Penggunaan Teknologi Informasi. Adapun target kami dalam waktu dekat ini adalah agar beberapa jenis pengujian yang dilayani Lab Penelitian Terpadu mendapatkan akreditasi ISO IEC 17025-2017. Dengan adanya akreditasi bertaraf internasional ini, diharapkan tingkat kepercayaan konsumen untuk melakukan pengujian meningkat, dikarenakan memenuhi beberapa aspek kualitas, antara lain ketidakberpihakan, kerahasiaan, keakuratan alat uji, kompetensi penguji, pemilihan, verifikasi dan validasi metode, serta pengendalian data dan manajemen informasi.
Kami anggota dari UPT Laboratorium Penelitian Terpadu USU siap menjalin kerjasama untuk membantu penelitian Anda, dan kami siap mengabdi pada masyarakat untuk memajukan bangsa Indonesia.
UPT Laboratorium Penelitian Terpadu USU.
Coba buat profesional.
Sugai sensei, ohayo gozaimasu.
Ohayo gozaimasu. Ohayo gozaimasu. 聞こえてますか? Can you hear me?
はい、はい。
はいはい。
よく聞こえます。
Okay, thank you Raisa and Veryani. Assalamualaikum warahmatullahi wabarakatuh. Good morning everyone. Ohayo gozaimasu, Sensei Professor Motoyuki Sugai from Antimicrobial Resistant Research National Institution Infectious Disease Japan. The Honorable the Head of Integrated Research Laboratory Rahmi, Karolina S.T.P. The Honorable the Vice Rector at 3rd USU, Professor Dr. Prof. Anjelisa Zaitun, Hasibuan, S.Si, M.Sc, APT. The Honorable the Speaker of the webinar Top Scientists this morning, Professor Motoyuki Sugai. The Honorable all lecturers, researchers, and students of Universitas Sumatera Utara, and the Honorable all participants of this webinar. I am Adrian Hilman, the moderator of this webinar. I'm very pleased to see you here and welcome all of you to webinar Top Scientists, with the topic is about Introduction of Antimicrobial Resistance AMR Research, focusing on people and the activities of the AMR Research Center. Ladies and gentlemen, our speaker today is Professor Motoyuki Sugai. He is the Director in the Department Antimicrobial Resistance Research Center, National Institute of Infectious Disease, Japan. He has received his Doctor of Dental Surgery in Dentistry at Hiroshima University 1984 and PhD degree in Microbiology at Hiroshima University 1987. He has a number of important positions in Japan and abroad, such as a Vice Director, Graduate School of Biomedical and Health Science, Hiroshima University, 2004 until 2012, Dean of Faculty Dentistry, Hiroshima University, 2012 until 2016, visiting professor of Department Antimicrobial Resistance, Graduate School of Biomedical and Health Science, Hiroshima University. Also, roster of experts in joint FAO and WHO expert meeting on foodborne antimicrobial resistance AMR 2021, head of WHO collaborating Center for AMR Surveillance and Research 2021 prison, and many more. He also has an H index of 52 in Scopus with the total of 233 articles. He is also often a keynote speaker at various international conferences and symposiums in Asia, Europe, and America. Before the presentation begins, let me inform that the presentation will be going on first. Professor Motoyuki Sugai will be invited to present the material at most for 60 minutes. After that, there will be the question and answer session. Now, allow me to welcome the speaker, Professor Motoyuki Sugai, to deliver his presentation. Professor Motoyuki Sugai, the floor is yours.
I would say good morning, everybody in University Sumatera Utara, and thank you for inviting me to give a talk about the AMR research project, especially running in our AMR Research Center. And I'd like to share the slide.
Can you start the slide, Professor?
Yes.
Okay.
It's okay. Can you see?
Yeah, clearly.
Okay. So today's my talk is about our challenges to stop AMR in Japan, because after staying about 20 years as a professor in the Hiroshima University as a professor of the bacteriology, then I moved to the National Institute of Infectious Disease in Japan, in Tokyo. So I moved from Hiroshima to Tokyo 3 years ago, and I was asked to establish the AMR Research Center in Japan. And so during these 3 years, I challenged several aspects of the countermeasure protocol in Japan, and I'd like to introduce some of them here. And now we are suffering COVID-19, and which started in the end of the 2019. And so compared to the COVID-19 AMR, I am using this slide very often. COVID-19 could be a tsunami. On the other hand, AMR is like rising sea level according to the change of the climate. And the COVID-19, it suddenly appeared, visited us, so it's suddenly noticed, and the virus is really pathogenic and virulent, and there is clear link between the virus infection and death. And also recently vaccine was developed, but still many countries are struggling with COVID-19. But when compared with the COVID-19 AMR, usually in hospital, it's going to spread without notice. So when you notice, already it's all over in the hospital. And most of the bacteria involved in the AMR are usually not harmful to the healthy, but virulent to the old or the have some underneath infection or some underneath problem, such as cancer or general health problem. And always this AMR the problem related to the AMR occurred behind the hospital. So most of the public people do not see the real problem of the AMR unless you enter into the hospital as a hospitalization or someone. And talking about the deaths related to the AMR, it's more complicated than the COVID-19 because most of the suffering people are very old. So it's very difficult to distinguish whether it is due to the AMR or due to some other complicated disease. So the causal link to death is sometimes questionable. And also, compared to the COVID-19, the AMR, the countermeasure of the AMR requires not only hospital, but also other sectors like agriculture or environmental. So it involves multiple sectors. So, and so I'm always showing this slide in the first of my talk. In Japan, after the declaration of the WHO Global Action Plan in 2015, here, because WHO, by that time, WHO didn't much think of the AMR problem worldwide. And so because of that consideration, they started WHO General Assembly here and held a WHO Global Action Plan that requesting all the participating countries to build their own national action plan. So in Japan, according to this Global Action Plan, 2016, we established a national action plan on AMR. So this action plan starts from 2016 to 2020. And in 2017, according to this national action plan in Japan, 2 institutions were established. The national institution was established. One is AMR, Clinical Reference Center, established in the National Center for Global Health and Medicine. This is a hospital in Tokyo, in Shinjuku. The other one is AMR Research Center. Our center is built in National Institute of Infectious Disease, which is also headquarters also situated in Shinjuku. But our center is present in the remote area of the Tokyo. Anyway, the 2 centers was established. One is mainly focusing on the clinicals. The other, our AMR center, is more to the basic research and also the epidemiology, surveillance. So today's talk about, most of the talk is about the surveillance in Japan, what we are doing. So every year we had a national meeting, Nippon AMR One Health meeting, and we published a report. And in 2019, the COVID-19 pandemic started. So we cannot finalize this national action plan in 2020, and still now extending 1 year. And we are expecting to start the new action plan from 2022, but at the moment we don't know whether we can really start 2022 or 2023. But we are now preparing. We are now look back our previous activity, and also we are preparing for the next action plan agenda. And this is AMR Research Center in Tokyo, but which is in the rural area of the Tokyo. This is the time I first came to this center in 2018, and you can see the snow. Even in Tokyo, in the rural area, you can see very rare case, but probably 1 or 2 days in a year, you will see a snowfall. So there's still remaining snow. And in Japan, we have as a National Action Plan 2016 to 2020, we have 6 goals set up: the public awareness and education, the surveillance and monitoring, infection prevention control, and appropriate use of antimicrobials, research and development, and international cooperation. Our center is mainly required to join this activity in the goal 2, surveillance and monitoring, and also the 6, international cooperation and research and development, goal 5. So 3 point our mission. So I will start with the surveillance and monitoring. So in Japan, related to the AMR, we have 2 national surveillance systems. One is National Epidemiological Surveillance of Infectious Disease. We call it NESID. So NESID is directed by the infectious disease control law. So this is mandatory surveillance pursuant to the Act on Infectious Disease Control Law, the mandatory reporting of infection cases. So what kind of cases? These are the notifiable disease, VRSA infection, VRE, MDRA, and CRE infections. And also we have the sentinel surveillance, that's MRSA, PRSP, and MDRP. And recently, from 2017, we started to check, especially check the CRE by using the PCR system. And sometimes we will use whole genome sequencing, but among them, in the future, we will conduct the PCR screening of the CRE, MDRA, and VRE. Fortunately, we don't have any VRSA infection in Japan. So the other surveillance is so-called Japan nosocomial infection surveillance. So this surveillance is called JANIS. JANIS is voluntary surveillance. The participation is voluntary. And in Japan, we have more than 8,000 hospitals. And among them, now over 2,000 hospitals are already participating in this surveillance. And contrary to the NESID, this is a pathogen reporting, so laboratory-based surveillance. So in the NESID, the case is only infection case. So if the patient becomes infection by the AMR pathogen, then you have to report. This is mandatory. On the other hand, this is JANIS system. This is a laboratory-based surveillance. So when the people go to the hospital, in the hospital, you will take the sample, and sample should be processed to identify the bacteria and also measure the susceptibility of the bacteria to the antibiotics. These information will be accumulated in the computer of the hospital. So by using this information, we will do the surveillance. This is laboratory-based surveillance. So information of antimicrobial resistance rates of clinically important pathogen are detected by this JANIS system. So the bacteria, including in JANIS system listed here, a lot of bacteria, probably you cannot see. So first, I like to introduce the recent activity of the NESID. In Japan, this surveillance started in 2014, so this is not accurate. But after 2014, 2015, 16, 17, the total number of the CRE infection in Japan is around less than 2,000. So less than 2,000 people are infected by the CRE. So this data coming from the local public health institute. Okay. And in 2017, we started a PCR investigation of the causative agent of this CRE infection. And in 865 cases in 2017, almost 30% are caused by the carbapenemase positive isolate. So carbapenemase producing such as E. coli or Klebsiella or such pathogen. And if you look at this 30%, over 90% of the carbapenemase type are domestic type. In Japan, IMP is the most prevalent carbapenemase isolated in the CRE. And the rest of them, maybe 5% or 8%, is NDM, KPC, or OXA48, which is imported from outside. So probably brought from the outside countries. Interestingly, since Japan is like Indonesia, we have the how to say long island from the north to the south. And if you look at the Hokkaido and the Kyushu area, the prevalence of the bacteria causing infection carrying this IMP type is different. For instance, Hokkaido area or northern area, you will see more often the Enterobacter cloacae carrying IMP. On the other hand, from Kinki or Osaka to Kyushu area, you will see more often the Klebsiella pneumoniae or E. coli carrying IMP carbapenemase. So even in the small country like Japan, you will see the different prevalence of the pathogen carrying carbapenemase. So this type of information is very important by using the PCR investigation. In Japan, IMP type, talking about IMP type, the major carbapenemase isolate in Japan, there are two types known. One is IMP1, the other one is IMP6. If you look at the prevalence, there is a clear difference. This IMP6 was only accumulating this Osaka region, Kansai region, but IMP1 is distributed all over Japan. So again, this type of information can be obtained by the PCR. So interestingly, only 5% of the carbapenemase producing CRE in Japan, compared to 2017 and 2018, there is clear increase of the occurrence of the CRE infection caused by this type of the imported type CRE. In 2017, this type of infection only occurred in the central area, like a Tokyo area or Kyushu, Hakata area only. But in 2018, this infection started to occur not only in the central region, but also the district, such as Okayama or other part of Japan. So this information is also very important to track.
Excuse me, Professor, your voice mute.
Oh.
Okay, thank you.
So from which, should I say again, this slide? Compared to 2017 and 2018, so there is a clear difference, and there is an increase of the incidence of the CRE infection by the imported case. So you can see the red color N. This means NDM type carbapenemase, especially by the E. coli. And this is coming from the other countries like China or the other Asian countries. So this increase is also show us a lot. Another important issue about this NESIT is not only detecting the infection, but also we have to take action. So this is a flow of the NESHID-based our action system. So if there some hospital has an outbreak of the notifiable AMR infection, then this information, this is notifiable, so this information should be go to the public health center in the local, and they will register. And also the pathogen should be sent to the public health institute in the local. And in the local public institute, they will do the PCR and also sometimes pulse field electrophoresis to compare the isolates sent to this institute. So these two information, so patient registration and pathogen registration, in every week in NIID, we have the conference of the risk assessment to see in which region of Japan is there any accumulation of the report of the such notified AMR infection. So if you find some accumulation or onset of the appear to be the increase of some isolation of the some pathogen, then we will contact the hospitals and also public health center and public health institute to see whether they taking really taking proper action against this. They really noticed about this increase of the infection. And in some case, we have the FETP, so Field Epidemiological Training program. So we will send the people to support the containment of that and take the necessary option to go to the hospital. And also in our AMR Research Center, we have the NGS machinery. So we will process the genetic or genomic information from the isolate. And this information is directly go to the Public Health Institute. And this information is used for the containment. Okay, so next one is the JANIS. JANIS system is, as I told you, the mandatory. And these two systems, JANIS and also NESHID, are all run by the Ministry of Health. And we are the running body of the JANIS. So JANIS has five divisions: clinical laboratory, and antimicrobial resistant bacterial infection, and surgical site infections, and intensive care unit, and neonatal intensive care unit. So these, the prevalence of the major MR bacteria and incidence of the MR bacterial infections, and incidence of surgical site infections, incidence of device-associated infection, and incidence of all infections in ICU are reported independently. So especially these clinical laboratories much more involved in the AMR research. So this is, as I told you, in Japan, almost 8,000 hospitals are there. And if you classify the bed size like this, then the participation number of the hospital at the beginning in 2007, very small, but gradually increased. And in 2015, so JANIS participation has become the prerequisite for reimbursement for the infection control. So the hospital get money by participating into the JANIS. This will accelerate the increase of the small number of the bed hospital. Right now, it is now in 2021, it is now over 2,000 hospitals are now enrolled. So this is the context of the participating hospital in Japan. So only very small amount, but over 900 beds, almost 90% of the hospital already participate in JANIS, 80% in 500 to 900 beds hospital, and 50% of the 200 to 500 beds hospital. But talking about less than 200, only 12%. So this is a challenge. Now it is increasing, tremendously increasing right now. And in Janis, every day the patient come and the sample was taken, and clinical laboratory participating hospital is able to submit the data as a Janis format. Actually, every machine, automated MIC determination machine, has some converter in Japan. Some already installed a converter from their hospital format to Janis format, data formatting. So this is key point one. And then this Janis format data can be compiled in the server of our center, MRI Research Center, and we can aggregate all the data together to prepare the data, the feedback report, and also national data. This is the number of isolates culture at the participating hospital. This is, I'm sorry, pretty old, but right now, about 8 million samples per year are incorporated in this Janis system. So how the data can be used? One is to generate an open report. So every year we generate the national report based on these data. And this is freely accessible. So everyone can see our national report. In Japan, what kind of the bacteria are prevalent, what kind of MR bacteria present. And also, we did the data analysis and send back the data to each participating hospital, their own report together. So in their hospital, they can compare the national report and their own hospital report to validate the infection control system in each hospital to benchmarking at their hospital. So which will motivate the infection control in each hospital, which is good. Okay, so this is right now the percentage of facilities reporting specific MR bacteria. For instance, MRSA is everywhere in Japan, so almost 100%. But fortunately, there's no VRSA report. And talking about the VRE, less than 10%. PRSP like this, MDRP like this. In such a way, this is based on national report. So CRE, around 60% of the participating hospital is experience the CRE case. So this is a summary. So in each hospital, receive this type of antibiogram. So national report is reported as an antibiogram. And you will see blue is susceptible, red is resistant, and yellow is intermediate. So each bacteria, you will see that each antibiotic susceptibility can be seen as an infographic. So in each hospital, they receive their own hospital's report together with the national data. So you can compare this and you can consider that how your infection control is working in your hospital or not. So this will be good benchmarking data for each hospital. So by using this Janis system, we can trace in each specific AMR bacteria trend. For instance, this is a trend of the MRSA. In Japan, the MRSA ratio is now decreasing, but now almost stopped and flat around here. On the other hand, fluoroquinolone E. coli and third generation resistant, cephalosporin resistant E. coli is increasing like this. So in such a way, by using this Janis system, we can we can understand the trend of the AMR pathogen appearance in Japan. And in Japan, one of the unique characteristic of Japanese National Action Plan is the government set up the numerical target for each pathogen. For instance, in humans, percentage of the penicillin-resistant Streptococcus pneumoniae, our target is less than 15% in 2020. Or, let's see, medicine-resistant MRSA, 20% in 2020. So this type of numeric target was set in and written in the National Action Plan. And there is a lot of objection occurred, but finally we are feeling that this type of the numerical target is very good for the motivation of the medical field, especially in the hospital. This is also set to the animal side. For instance, percentage of tetracycline-resistant E. coli should be less than 33% in 2020. Third generation cephalosporin-resistant E. coli, same level as G7. Fluoroquinolone-resistant E. coli, same level as G7. This is for the animals side. So animal side and human side, they set up the numerical target. And I am in charge of the human side. And this is a result of the, our 5 years follow-up of this numericals. As you can see, some of them are like, which one? For instance, Pseudomonas aeruginosa carbapenem resistant, now reaching to the target, but others are still beyond the target. And CRE is already in the target. In such a way, we can figure out, not only we, but also everyone can figure out what the situation we are right now by using this type of target, numerical target. So probably I think that in the next action plan, we will also employ this targeting system, numerical targeting system. And another important trial in Japan during this National Action Plan 2016 to 2020, we are generating the integrated database. So this is a integrated database of integrating the several databases into one-stop report based on infection control law. Like this is NESID, and this is a JANICE, and this is Japan nosocomial. This is the Japan antimicrobial consumption surveillance. This is about the antimicrobials. So data of the antimicrobials, and they are also including the sales of the surveillance, sales of the antimicrobial surveillance. So they will integrate all the surveillance in one stop and create the integrated surveillance system database. And this is called Japan Surveillance for Infection Prevention and Healthcare Epidemiology. So by using this system, in one stop, you can see the trend of the MR pathogen and also antimicrobial use and also infection control index of each participating hospital. So by grouping the hospital, for instance, in the regional hospital, they can share these data together and they can use these data to the antimicrobial countermeasure in each region. So in Japan, the regional countermeasure is very important, we believe. And so for that purpose, the government started to set up this integrated database. This is called JSIFE. This is now starting, and already more than 400 hospital groups are involved in this JSIFE and start to use this information. Another thing about this National Action Plan, we are able to combine the data of the JANICE system, this is the human side, and also JBARM, this is the animal side. This JBARM is run by the Ministry of Agriculture, and their data are now combined to see you can compare the data of the human side and animal side. This is reported in Nippon AMR One Health report. So this report is started to in 2017, and every year we publish this report, not only Japanese but also in English. The one good thing through this One Health report, we came to know that in the broiler, in the animal side, previously in Japan, the broilers for the hatchery, they use some cephalosporin to prevent the bacterial infection. So at that time, the rate of the resistance of the ESBL from the broiler is very high. But after noticing the problem, they stopped to use the cephalosporin in the broilers. And then after that, the prevalence of the ESBL in the broiler is sharply went down. Instead, human side, there's no change. So this is one of the indication that the aspect of what is going on in humans and what is going on in the animal side are sometimes distinct. On the other hand, we have another contradictory data of another aspect. So it's not by using this data, we cannot say the human side and the animal side are completely different. We cannot say by using this data, but the thing is more complex. But in this specific site, it's good to see that by using the comparison of the human side and animal sectors in one stop, One Health report, that's a good thing. And we also have some challenge about the surveillance. One is Japan antimicrobial resistant bacterial surveillance. This is called JABS. As I told you, we have already have the long history of the JANIS system. But the JANIS system doesn't have genomic information because they don't handle the bacteria itself. They only use susceptibility data from the hospital. And so we started JABS from 2019. The other one is a WHO tricycle surveillance. This is started in 2020. This is using the ESB producing E. coli as an indicator strain, and the surveillance was taking place in human, food, and environment. This is another our challenge. So first, I'd like to introduce the JABS. So JANIS data has only the ID, the species of the bacteria and the antimicrobial susceptibility testing results. This is the only data used by the JANIS. But there's no pathogen information such as gene or genome. So to supplement this, we started the AMR bacterial surveillance, JABS. So JABS handles the resistance genes, virulence genes, plasmid profile, multi-local sequence typing, and genome data. And this is a scheme of the JABS. So we are focusing on some of the AMR bacteria such as ESBL producing bacteria, CRE, VRE, Acinetobacter resistant to carbapenem, Pseudomonas aeruginosa resistant carbapenem, or MRSA. And we are collecting bacteria from the hospital already participating in the JANIS. And JANIS system, already 2,000 hospitals are already participating. And we choose 200 hospitals from the JANIS system. And we collected a sample from each hospital and did the genome sequencing and also susceptibility testing to accumulate the data to deposit in the AMR bacteria bank. So through this system, we need to improve the high throughput genome sequencing protocol. So we employed a robot and also genome sequencer. and also some semi-automatic robot for the processing of the PCR. By using this system, we are now able to handle 2,400 genomes per month, and also 18,000 samples of the PCR per month. So these two high throughput systems work very well for the genomic surveillance in Japan. So I'm sorry, this is in Japanese, but this is the flow of the process. So taking sample and do the PCR by using automated electrophoresis system. And then data will be accumulated in the Excel file. And the first screening will send to each hospital by like this Excel format. And for the next genome surveillance, the library production and the NGS were done in-house. And the data will be deposited and after that analyzed by our informatician. So this is one challenge in our MR Center. The other one is a WHO integrated global survey of the ESBL producing E. coli using One Health approach. So surveillance in humans from the isolate from the bacteremia and also from pregnant human rectal swab. This is as a healthy people and diseased people. And the surveillance from in the food chain is chicken, caecum, ESBL, and surveillance in the environment, liver and sewage ESBL are taken and compare. So during this surveillance, we are receiving large amount of the pathogen from nationwide. And also we are imported from US CDC as of the AR panel. And these are accumulating in our bacterial bank, AMR bacterial bank. And storing not only the bacteria, but also the sometimes clinical information and also genome database. So by using this database, we were trying to create the AR panel. So representative AR bacteria. And this will be distributed to the hospital for the quality control and also pharmaceutical company to generate a new antimicrobial drug against the AMR bacteria, and also the research to the academia. So this is a very important mission for us from now. From next year, we are starting the distribution of the AR panel to the public. And also we are willing to share the information with other countries' AR bank for the international collaboration. So last part is about our trial for the international collaboration. And talking about the surveillance, in Europe, they have a very classical and also very important sophisticated surveillance system. EASnet cover the European countries, EU countries. And WHO also started large surveillance in the northern part of the Europe and Russia. It's called CESAR. And we have Janice. And also in China, they have the CARS and also CHINET. And in Korea, they have Korean grass. And in Australia, they have the APAS. And in America, there is North America, US CDC, and South America, Labula is using their surveillance system. But the positive present in this Southeast Asian countries. So, but most of the our importing important AMR bacteria are coming from these countries. So we are wondering whether we can contribute to the establishment of the surveillance system in these areas. So we are now, because Janice is a very economical surveillance system, you don't have to buy a big server. we can use cloud system to pay less and also can get a very good efficient data. So by using the Janus system, we created the English version of the Janus system called AsiaSnet. This is backed by the Ministry of Health, and the maintenance of the cloud system of the AsiaSnet is now backed up by the Ministry of Health. And we are in collaboration with the World Health Organization and other sectors to establish, and also we are hoping to be used in the Asian countries. So this system is using cloud system, is very highly secured, like a secure system by the such as IBM or McDonald's or Apple, same cloud system we are using. And by using this system, each participating country can aggregate their participating hospital data by using this AsiaSnet. And this is very highly secured. So other country cannot see other countries' data. So this is very highly secure system, AsiaSnet. And we are now promoting to be used in other countries. And now in Thai, Vietnam, and Mongolia, we have already established an MOU, and some trial is already going on. Unfortunately, due to the corona pandemic, our system is now interrupted, but we are now waiting for the restart, especially in the Vietnam. And also talking about Indonesia, we were in process of establishing MOU with East Java province and LOA with Dr. Sotum Hospital with the help of Professor Kuntaman and other important figures in the AMR countermeasure in Indonesia. But unfortunately, corona pandemic stopped these activities, and we are now eager and waiting for the restart of this establishing MOU and start the AsiaSnet trial in Indonesia also. So these are our activities during this National Action Plan 2016 to 2020 in our AMR Research Center. Of course, our trial is a collaboration with the Ministry of Health and also the NCGM in the another side of the AMR Clinical Research Center in the Shinjuku Hospital. And I think I will acknowledge all the members here for this activities. So, terima kasih. Thank you very much.
Thank you very much, Professor Motoyuki Sugai, for giving us such informative and perfect presentation of antimicrobial resistant research and introduction your institution to us and over the cooperation with the other countries. And now come to the question and answer session, Professor. Could we?
Yes. Okay.
I like to have several questions from participants in chat box. I will check first. Okay, this is the first question, Professor, from Febri Rizky. Good morning, Professor. I want asking 3 questions. What's molecular method or other method to identify case of CRE infection or all pathogen for this research? And how to solve many obstacles related to data for this research? I think this research collect lots of data, and is this research use machine learning method to predict lots of data? Thank you, Professor.
So the most important method, molecular method, is a PCR. It's because of the in the local hospital or local health institute, they don't have much instrument. So and the handy instrument is a PCR machine. But even in Japan, most of the hospital didn't have the PCR machine by that time before corona pandemic. But due to the corona pandemic, the necessity to do the PCR in each hospital increased. So they purchased the PCR machine. So now almost all the hospital in Japan somehow has settled the PCR machine. So which is good for the surveillance in the MR research and MR surveillance, and everyone can use PCR. So PCR to detect the carbapenemase or some important indicator of the MR pathogen, which is good, but it is just a first step of the surveillance. If you want to compare the bacteria A and bacteria B to see whether the relation between the bacteria A and bacteria B, you need another system like a pulse field electrophoresis or other system to see the similarity of the two bacteria. For that purpose, now it is used a PFG, pulse field gel electrophoresis system, but now it will be converted to the whole genome sequencing. Very soon we will compare the data by whole genome sequencing. But still now it's very expensive, but the price is now immediately going down. So by using outsourcing the company, now in Japan, the price of doing the PCR and doing the whole genome sequence is almost comparable. So the next generation, I'm pretty sure that the whole genome sequencing is a must in the surveillance system. Yes.
Okay, thank you. Febri, how about your question? Is it all of your answer? Do you want? Okay, thank you. Yeah, thank you.
It's clear.
Okay, yeah, thank you. And please, everyone in the room, you can ask the question to the Professor Motoyuki Sugai directly. Please open your mic. Is there anyone? Maybe Professor Motoyuki Sugai, I want to ask you, it is a simple question. Does NIID stand under the Japanese Ministry of Health? And is the system you and team in NIID built related to Society 5.0 in the health sector? Because it is interesting system.
You say again?
Yeah. Does NIID stand under the Japanese Ministry of Health?
Yes. Yes.
And is the system you and the team in NIID built related to Society 5.0?
Yeah.
In the health sector.
In university, the Society 5.0 is very prevalent, but I don't exactly don't know about the Society 5.0.
Okay. Okay, it's okay. Okay, anyone in the room wants to ask the question to Professor Motoyuki Sugai, please? Or maybe, Professor, if we see from your slide presentation from start and the end of the slide, how to start and build a system to link research in universities with facilities provided by the government. By your experience, your start to build.
When I was in university, I was very much interested in the local. So I was in Hiroshima. So like, I don't know whether in your country, but in Japan, the local district, medical district is very important for the MR countermeasure. For instance, in Hiroshima University, the only Hiroshima University is the national university in the Hiroshima region, and Hiroshima University Hospital is taking responsibility and lead the Hiroshima region's MR countermeasure. So we use a research grant to purchase the PCR machine and also NGS machine. And in collaboration with the hospital in the Hiroshima region to start collecting the sample every year and accumulating data. So this data is very useful for the countermeasure in the Hiroshima region. So this types of the regional network is very important. Not only university, used to include involve the hospital, participation of hospital. You may, in our case, I created such kind of the consortium, inviting the hospital and also the some governmental institute. Also, we invited to include. So every how to say members of the important figures in this region we included, and to create a one sector discuss about the AMR situation in the Hiroshima region. So this system is very important, I think. So this regional countermeasure is very important from the point of the national AMR surveillance, yes. So for the university, I think, I don't know about the Sumatra, but your university will take responsibility of your region to see and also monitor the AMR research, through the AMR research to AMR situation in your region, which is very important. And you will probably has a contact with the central government, and this information will be also useful for the Indonesia.
Okay, Professor, we realize in maybe in Universitas Sumatera Utara, the MRI research is not a primary focus of the topic, but I think after this webinar, we will conducted the MRI research to our lab in the laboratory. And could it be possible for us to start this research in Indonesia? Because you know we are in the tropic climate, and there are many endemic of microbials in different regions. Is it possible?
Could you say again? I cannot. The conversation was stopped sometime.
Okay. Maybe this is because the signal. Okay, if we want to start the AMR research in our university, especially in integrated research laboratory, is it possible for us in Indonesia to do as you did in Japan by isolating microbes in different area? Because you know we are in the tropic climate, and there are many diversity about the microbial in here. Is it possible?
I think so.
Okay.
I think so. You can do that. And I have the colleague in the Airang University. They are also collecting the sample in the Java, and also someone is collecting sample from the Bali, from the many sectors, like not only human, but also from the rural country and from the animals and the sewage. Many One Health for the One Health research, it's possible, I think.
Okay, thank you, Professor. Is there anyone to ask the question for Sensei or Professor Motoyuki Sugai, please? You can open your mic directly.
Sorry, Sugai Sensei. I might have a question for you, but I might need to explain this a bit. Okay. Now I've just seen the map of Chinese participating hospitals, and I think the question is this: do you think the infrastructure of hospitals in Japan are all the same? Because when I'm looking the location of the participating hospitals, some of them seem to like they have no participating hospitals such as like Wakanai or Oita, something things as such. Do you understand my question, Sensei?
I cannot understand. Could you Explain again.
Okay, I'll try this again. Okay, so I've seen the map of Janis participating hospitals.
Yes.
And because I seen their places that don't have the participating hospitals, such as like say Wakanai or Oita.
Wakanai
So
is in Hokkaido, and Oita is in Kyushu. Yeah.
So I think so. My question is this: is hospital infrastructure in Japan is the same for all 47 prefectures?
Infrastructure means are they using the same type of the machine for the detection of the bacteria? Are you saying that mean?
Yeah.
Yeah. In Japan, several types of the machine for the identification of the bacteria you can purchase. And in Japan, due to at the beginning of this Janis system, with a discussion with Ministry of Health, every type of the machine in Japan will install the converter from the each hospital information to the Janis format system. So even if you buy different types of machine, which is already installed this converter system, so by using any type of the machine, you can generate the same format of the for the Janis system, which is very good, and uniform information can be obtained through all over Japan. So even Wakanai or Oita or Okinawa, even Okinawa, we can get the same information, same level of information. But one thing we are thinking it is a problem is when you compare this machine of the different company, there is a difference in the how to say activity to quality. So we need to raise the quality of each company to comparable to obtain more informed data for the good data for the analysis. That's our challenge right now. Quality control, yes.
Okay, Sensei, thanks for the answer. Arigatou gozaimasu.
Domo.
Okay, thank you. Okay, is there anyone? Okay, please, maybe this is the last person to ask to Professor Motoyuki Sugai. Professor, Dr. Gigi Rizka Hidayati. Please unmute voice, okay.
Okay, thank you for the chance that you have given to me. Ohayo gozaimasu, Sensei.
Ohayo gozaimasu.
How are you?
Fine.
Sorry for disturb you for many times because my email to you.
No problem.
I want to ask you some question. The first is is it very interesting if someday we can collaborate with NIID, especially to examine some samples, right? I want to ask, are there any special requirements for the samples that can be accepted and examined in your laboratory? I mean, at NIID. And the second, as your experience, are there any samples that can be rejected by your laboratory? If there, why some sample can be rejected? And the last question is, I'm so interested about the employers in your laboratory. How to recruit the employer in your laboratory? Maybe some of Indonesian people can be entered to your laboratory as your employer. Okay, I think that's all of my questions. Thank you very much.
Thank you, Rizka. And the first question about bacteria sampling collaboration, and we are always open to collaborate with other countries. And to analyze the sample in NIAID, if you want to send us living bacteria, there is a very strict regulation in Indonesia to send overseas the sample, maybe due to the Cartagena regulation, and we need to clear that part. That's a point. And from Japan side, some fungus and some Pseudomonas are prohibited to import from outside because of the agricultural matters. So that type of the most of the fungus are requested to report. So even reporting some of them we cannot import from outside. Otherwise, most of them are easy to be shipped from outside countries. And for the employer, we have two types of the employer. One is the formal researcher, and you need some guts and also willingness to work in NID, and also with some publication. And the other type is non-permanent researcher, which is 1 year slot, and that type is much more easy to apply. And also by using some scholarship, you can apply to spend 1 year or 2 years, like you, more shorter time to spend to work together. So there's several options. And also recently WHO or Framing Fund, maybe Framing Fund or other funding system will propose some overseas researchers to support. So by using such a scholarship is also good for the application. So thank you.
Okay, thank you, Prof.
Okay, thank you, Dr. Gigi Rizkaidayati. I still have a time, Professor Motoyuki Sugai, because in the chat box we have one question left. Can I read for you?
Yeah. Yes.
Yeah, okay. Good morning, Professor. I want to ask, is there anything that specifically triggers the appearance of AMR in bacteria? And is it possible that in the future, more antibiotics discovered, more AMR bacteria will also increase? What is the cause, Professor?
Yeah, that's a good question. And if you use antimicrobials, there is always the resistant to this antimicrobials appears. This is there's no exception so far, as far as I know. So you don't use the antimicrobial is the best solution, but when you become sick, you cannot do that. So we have to use antimicrobials. So our message is to the prudent use of antimicrobials. So not to use excessive amount of the antimicrobials, and useless antimicrobials you don't have to use. So this type of education is very important for the public. In Japan, during this National Action Plan, by using commercial, we pronounce that this importance of the prudent use of antimicrobials to the public and also medical field. So in the medical field, it worked, and now the total use of the antimicrobials in Japan, especially the third generation cephalosporin, is now decreasing, which is good. But for the public, still most of the people in the public don't understand that take the antimicrobials and antibiotics when they become flu. So they don't really understand, they don't really understand the difference between the influenza and bacterial infection. So education is very important. And I think from the very young children, you need the education about the AMR. That's a very important point. Thank you.
Okay, thank you, Professor, for your excellent answer. And ladies and gentlemen, we finally come to the end of this seminar. And the last, I want to thank the speaker of the informative and excellent presentation for Professor Motoyuki Sugai, and all the participants for the very active participation this morning. And maybe I will give the, we give the applause for the speaker and for you all. And then I will return the event to Raisa and Veryani. Maybe Miss Veryani can speak with Professor. Thank you.
Thank you, Mr. Adrian. Finally come to the end of today's agenda. I would like to say thank you.
お時間となりますので、これでウェビナーを終了したいと思います。
I would like to say thank you to our keynote speaker, Professor Motoyuki Sugai, for the very interesting, informative presentation. We learned so much. Arigato.
Thank
you very much.
菅井先生、大変参考となるお話をありがとうございました。
はい、どうも。
本日はお忙しい中、スマトラ大学のウェビナーにお越しいただき、誠にありがとうございます。
はい。
Also, thank you to all the participants for joining us today, and for Miss Veryani, thank you. And last, I would like to apologize for any mistakes happened during this webinar. And for any further information, you could follow us on our social media, Lab Terpadu USU. Once again, thank you very much. See you on the next webinar series from us. And for the certificates, we will tell you later. Inform you later. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Arigatou gozaimasu, Professor.
Thank you.
先生、お元気ですか？
はい、元気です。
先生は今どちらにいらっしゃいますか？東京ですか？
東京です。東京です。
はい。そうですか。
日本語上手ですね。
いえいえ。東京の天気はどうですか？もう寒くなりました。
だいぶ寒いですね。紅葉がだいぶ綺麗になってきました。はい。
はいですね。紅葉が。はいはい。ありました。Ini ada yang mau disampaikan?
Rizka, bye bye.
はい、先生、本当にありがとうございました。
はい。
ありがとうございました。
はい、どうも。
Makasih banyak Ines, Pak Adrian, Raisa, Wakil, Febri. Terima kasih semuanya.
Sama-sama, Bu.
Hai Raisa, Kak Rahmi.
Sama-sama.
Sama Desta.
Pak, izin yang jadi host Pak Ismet atau Pak Sentosa nih Pak?
Langsung mahasiswanya aja tadi.
Mahasiswanya aja, baik Pak.
Kalau dia share screen ya.
Oh iya, ya Pak. Saya izin keluar ya Pak.
Oke, ya.
Ya Pak, Assalamualaikum.